Hello everyone, welcome to week 2 of the course. This week we have an awful lot of material to cover. We are going to be covering 4 chapters in your textbook, so you'll want to be starting right away this week. First off, we're going to talk about DNA and the different structures that it can take on. Now, while DNA is always going to be a double helix, that double helix can be in the form of a linear molecule, or it can actually be a circular molecule too. And depending on the organism or the location, the DNA is going to take on a different shape. DNA gets organized with proteins, and it also gets organized into structures that we call chromosomes. So we're going to be looking at that, and then we'll talk more about how the DNA is actually used to make the proteins that are going to be operating inside the cell. We'll look more closely at the process of transcription and the various enzymes that are involved in that, and then we're going to look more closely at translation and how the information in the mRNA molecule gets converted into amino acid sequences. Once we've done that, we'll look closely at the process of gene expression and all of the steps that are involved in gene expression. Our assignments this week are going to be pretty similar to what you had last week. You will have a discussion. In that discussion, you're going to be investigating more about DNA molecules and the various shapes. You'll talk about the advantages or disadvantages of being circular in shape or linear in shape. And then you'll also have a couple different worksheets that you'll be completing along with a PCR lab.